Yes, we can.
Yeah.
Okay. So during the S phase of the cell cycle, the cell undergoes replication where it duplicates its DNA, and then thereafter there is mitosis during the mitotic phase where it divides into two daughter cells. Today, transcription in some regards resembles replication, but the fundamental thing is that we are running a photocopy. So let's say I have an image on my PC and I want to show you that image, but you can't see my PC screen. So what do? I connect my PC to a printer and then I print out the image for you and then I show it to you. So you are going to see a manifestation of the picture I have on my PC. So you can call the picture on my PC the blueprint, and you can call the picture I have given you the transcript of the blueprint. Another example is, I know before you all came into GAMP, you had to go to your various universities where you had your undergraduate education. You requested for your transcripts. So your results are actually with the academic affairs unit. What they gave you is a copy of your results. So it's a transcript of your results. So even today, if I should go to your university, if I should go to the academic affairs office, I'm going to get access to your results. But what I won't get is what they have in their computer. They would only give me a copy of what they have in their computer. That is the same thing with cells. Cells have an information, which is the nucleic information or genetic information. This information is kept very safely. Trust me, the cell does a lot to make sure that the genetic information is very protected. So in order to express the content of that genetic information, there's an elaborate system of transcription that allows the cell to benefit from the information through the transcription machinery. Now, the most important thing on this slide is the fact that magnesium and zinc are used in transcription. And then also this one. The fact that magnesium and zinc are used in transcription. And then transcription has three phases: elongation, initiation, and termination. That is all you have to pick up from this slide. This one, you get to follow as we go on, the 3' nucleophile and all that. But these are the three things you need to get from this slide, that it comprises initiation, elongation, and termination. It uses magnesium and zinc. And then the 3' hydroxy group acts as a nucleophile. The nucleophile is an electron donor, but there'll be more on that later. The next thing is that, yes, of course, transcription is not the same as replication. There's a difference. The one main difference is that whereas replication involves copying the entire genome or the entire set of chromosomes, in transcription, you're only copying a small portion, which is a gene or a cistron. A gene is the same as a cistron. So you get to hear polycistronic and monocistronic very soon. But the most important thing to know is that in replication, we are only copying a part of the chromosome. So it's less tedious. All right. So that is point number one we have to know from here. Point number two we have to note is that there's a template required. There's always a template required for transcription to go on. Just like replication, the polymerase has to look on something. By looking on that something, it transcribes what is required. Now, this is very important high yield point. So you have to be able to remember that. I say that there's an ant in my CNS. ANT, antisense non-coding template. And then you have C, N, and then S, coding non-template, and then sense strand. You have to know which is which at all time. But mostly, we always use template, non-template, template, non-template. So it's very important that you differentiate that. The template strand is the strand that the polymerase enzyme is directly producing complementary DNA for. The non-template strand is the strand for which the transcript is going to resemble directly, with the exception of thymine and uracil. All right. This is also very important. By convention, the regulatory sequences that control transcription are designated by the sequences in the coding strand. Please, if you don't understand this, let me explain. If you do, then we can go on. But this is a very important sentence. By convention, the regulatory sequences that control transcription are designated by the sequences in the coding strand. All right, we go on. Now, during replication, the entire chromosome is usually copied, but transcription is more selective. I already mentioned that. Only particular genes or groups of genes are transcribed at any one time. And so some of the portions are never transcribed. In fact, when you look at the fraction of the DNA that is transcribed, you wonder, so why did God give us so much DNA? Because you transcribe less than a significant portion of your DNA. The majority of the DNA left comprises of introns, short tandem repeats, enhancer sequences, promoters, transcription factors. But when we talk about genes that have to be transcribed into proteins, they are not so significant. So transcription, as I said, is not as tedious as replication for the cell. The last point here is also important. Specific regulatory sequences mark the beginning and end of DNA segments to be transcribed and designate which strand duplex to be used as templates. This is connected with the previous slide here, this very last slide here. So I believe it's self-explanatory. All right. All what I have said you can see in this diagram. And so by the time we move from this diagram, you should be expected to familiarize with all of these things on the left-hand side. The coding strand. Remember that the coding strand is called the coding strand because it doesn't have any interaction with the nascent mRNA. It just sits there idle. But at the end of the day, the nascent RNA looks just like it. So it's like in computer coding, when you use a code to transcribe a message. So at the end of the day, this RNA that is being produced here will look just like this coding strand. But this coding strand didn't have any contact with it. In fact, it was the non-coding strand that had contact with it. So that is why it's called the coding strand. It is coding for the mRNA that's going to come out. The same reason why it is called the non-template strand, because it is not a template. This strand here is the template. Another reason it's called the sense strand is that this strand can produce, if you want to, sorry, is it, okay, I have to admit someone, just a second. Okay, let's come back here. Yes. And it is also called the sense strand, as I was saying. The next thing, of course, the non-coding strand is the template strand. The transcription bubble, very important, is the bulge created by the RNA polymerase as it melts, it locally melts the double strand conformation of the helix structure. So this is the bubble. I used to think, oh, it's a bubble is just a description, but it's actually a technical term that you have to apply. For instance, if you're talking about the open complex, you can't talk about the open complex without mentioning the word bubble. I find it quite funny, but it's true. So you have to know that this is a bubble, and the bubble moves as the polymerase enzyme moves along the strand. The next thing, yes, is the open complex. The open complex comprises all the enzymes and factors that are responsible for getting the double helix structure melted and opened. Remember, in replication, these were DNA and then the single strand binding proteins. Over here, we don't have those. Over here, we just have the sigma factor and the general transcription factor in prokaryotes and eukaryotes, respectively. Can we move forward?
Yes, please.
Okay. The next thing is the nascent RNA, which is this one, the green one coming out. So as the polymerase enzyme moves in this direction, the nascent RNA falls backwards in that direction. Hello?
Hello.
Oh, okay. I think I went offline for a while. Did someone call me? Hello, is someone trying to call me?
If there is, I'm sure the person is probably trying to get in.
Oh, okay, I see. Okay, I've
Yeah.
let the person in. That's quite strange. Yes. Okay. So we move on. Also, it's very important. Um, I hope you can still see my screen.
No, please.
Oh, okay. Okay. So this is where we were. And so the next thing we're to notice is the unwinding and rewinding. So up front here, you are going to have unwinding. Up front here, you're going to have unwinding. That is right here. The DNA will be unwound by helicase activity. And then at the rear end, it will be rewound. And of course, there's topoisomerase activity over here where there's a supercoil, a positive supercoil. You need topoisomerase activity, just like in replication. Then the components of the RNA polymerase holoenzyme. The holoenzyme comprises the alpha subunit, the beta subunit, the beta prime subunit, the omega subunit, and then the sigma subunit. But there'll be more on that also, I think, next slide. Yes, this is a very important slide that you have to appreciate moving forward. There are four things here which are very crucial. The first thing is the promoter sequences, the -35 sequence, the -10 sequence, and the termination signal. These are three very important things. They are not part of the mRNA that will be transcribed out of here. They are just there. And this is what I was talking about in the fourth slide. That when you hear these sequences, they are being with reference to the top strand. So when you hear Tata box, it means that it's actually ATATT on the template strand. So by convention, the Tata box, the GACA box, all of them, they are mentioned with respect to the non-template strand or the coding strand. You have to know that. And they are the promoters. Call them the promoters or the promoter region. The space in between the promoters, 35 and -10, is what we call the linker sequence. It's also an important fact you should know. If the linker sequence changes from between 16 to 19, it can affect the rate of transcription. So everything here has to be perfect for the +1 sequence to be the initiating point. Now, this is the coding region, which we call the open reading frame. In translation, also, we have the open reading frame. So this is where the polymerase will start transcribing from. So polymerase will land here and then start transcribing in this direction. In fact, polymerase will land all the way from here, actually. But this is where it starts transcribing from. So all of these are just guiding it. This one will tell you, hey, you are quite close. Okay, then it's moving. Then it gets here, it says you are very close. Start counting from 10. So -10, -9, -8, -4, -3, -2, -1, 0, +1. So this is the +1 sequence. That is why it's called +1. So immediately it gets to +1, it starts synthesizing ribonucleotide monophosphates. In the termination sequence, there are two types. We have the row-dependent termination and the row-independent termination. But once you see a sequence here, it means that this is talking about row-independent termination. Its aim is to make the mRNA form a structure like this. Once the mRNA forms a structure like this, it will have no option than to fall off because the RNA polymerase does not know how to handle such a shape. It can only handle a linear mRNA structure like this. It can't handle this. So once the mRNA assumes this shape, the RNA polymerase will have to dissociate from it and then let it go. So the sequence over here allows the mRNA to form this shape. We'll come to that when we are talking about termination of transcription. Now let's focus specifically on prokaryotic transcription because it has special factors and special things that need to be talked about. The first thing is that in prokaryotic transcription, there is no post-transcriptional modification. Secondly, transcription can be coupled to translation, meaning that as transcription is going on, translation could also be going on from the 5' end of the nascent mRNA. And you find something very amazing that the tRNAs, that is the RNAs responsible for converting the mRNA, for reading the mRNA and synthesizing amino acids, coincidentally reads from 5' to 3'. Nature is quite amazing. So the mRNA is synthesized from 5' to 3', but then the tRNAs read the mRNA and synthesize amino acids from the 5' end of the nascent from the mature mRNA to the 3' end of the same mRNA. Now, the next thing you have to know about prokaryotic transcription is that they have polycistronic mRNA. Polycistronic mRNA. One mRNA could code for several proteins. Now, this is quite different from alternative splicing. Because you may think that at the end of the day, they are all the same thing, one mRNA coding for different proteins. But when we say polycistronic, it means that one mRNA is containing information from more than one cistron, from more than one gene. So from the name, you can tell what it's trying to say. Polycistronic mRNA. One mRNA comprising of information from more than one cistron. Of course, prokaryotic transcription is catalyzed only by RNA polymerase. That's all. So if you see in any question, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, instantly your mind should tell you that we are not talking about prokaryotic replication. We are talking about eukaryotic replication. Okay, what do you have to know about polymerases? Just like DNA polymerases, they synthesize from 5' to 3', meaning that they read the non-coding strand from 3' to 5'. They require a template. They are able to synthesize de novo, unlike DNA polymerases that need a primer. But their shortcoming is that they have a relatively lower fidelity compared to DNA polymerases. I think because they lack the 3' to 5' exonuclease activity, so they are not able to edit their synthetic activity. Next, they are processive, but not as processive as DNA polymerases. Why do they have to be? Why don't they have to be as processive? Because they don't have a beta clamp. They don't have the processivity factor. The clamp that is able to bind. The clamp that is able to clamp. Let me use the correct word. There is something that binds, but there is nothing that clamps. So I'm saying again that RNA polymerases don't have a clamp, but they have something that binds. But that binding is not enough, like the clamp, which you find in beta-2 clamp or the pCNA. So because of that, they are not as processive as DNA polymerases. Also, there's no rush because they are not synthesizing the whole of the chromosomes. They are just replicating a part of the chromosome, which is specific genes. So they don't have to be as processive as DNA polymerase. Now you have to know that the active site of the RNA polymerase is the point where the beta and the beta prime subunits meet. Where they meet is the active point. So let's say this is the beta subunit and this is the beta prime subunit. At the point where they meet, the middle here, that is the active site or the catalytic site of the RNA polymerase. The alpha subunit just holds the beta to the beta prime and holds the beta to the beta prime. So there are always two alpha subunits. Okay, that was what I just talked about here. So the alpha, anchors or binds the two together. This is very important. The beta forms the phosphodiester bonds, also very important. Then the beta prime is what binds. Remember I said there's binding, but there's no clamping. The beta prime does the binding. The omega is responsible for calling the subunits to come together. So it recruits. The sigma factor is the most high yield of all of this. So if you forget anything, please don't forget the sigma factor. The sigma factor is the high yield point. So I'm going to highlight that sigma factor is high yield. It does two things, but the main one is promoter recognition and binding. So if the cell wants to transcribe proteins that will metabolize nitrogen, then the cell will look for a sigma factor that can look for nitrogen metabolizing genes. So it will take DNA polymerase alpha, beta, beta prime, and then specifically bring a sigma factor that can transcribe genes that are responsible for nitrogen metabolism. So remember that when you have two of the alphas and the betas, is the core enzyme. For you to have a holoenzyme, we'll get to see it shortly. So this is the core enzyme. it's also important to know the core enzyme. The core enzyme is 2 alphas, 1 beta, 1 beta prime. The next important thing is that once it's called at the core, it means that it doesn't have eyes. So it can't synthesize what you want it to synthesize. It just does things anyhow. So as I mentioned earlier, the sigma units are the eyes. You see the sigma looks like eyeballs. So the sigma units are the eyes of the RNA polymerase core enzyme. So when the sigma factor joins, now the RNA polymerase can see well and locate the promoters. Once it finds the promoters, it can transcribe like no one's business. The next thing is that depending on the genes, that are required, where are we? We are here. Yes, this is a very important point. Depending on the genes that needs to be transcribed, you have to know these 3 sigma factors: sigma 70, sigma 32, and sigma 54. You know that 5 plus 4 is equal to 9. 9 for nitrogen. So if you want to synthesize nitrogen, you need 5 plus 4, which gives you 54. If you want to synthesize housekeeping genes, what are housekeeping genes? These are genes that the cell needs for normal metabolic activity, to stay alive. Like we human beings, to stay alive, we need to breathe, we need to drink water. Yes. So the cell also has certain genes that in order to stay alive, it has to transcribe. So those cells are called, constitutively expressed. They are always being expressed. They never rest. So when the cell wants to transcribe those genes, it uses sigma 70 as the eyes. If it wants to transcribe genes to enable it to respond to heat shock, let's say it's in a place where there's excessive heat and it wants to be able to respond, then it uses sigma 32. This is a high yield point, so you have to keep this in mind. Then we come to eukaryotic transcription. Is there any question up to this point before we enter eukaryotic transcription? Hello?
Hello.
Yeah, is there any question?
Yeah, please. I have a question with regards to the sigma factor. Hello.
Yes, what's the question?
Hello. Yes, please. I have a question. Can you hear me?
I can hear you.
Okay, please. I have a question with regards to the sigma factor. You said without it, the polymerase is blind. Doesn't know where exactly to start transcription from.
Yes.
So I want to know if it recognizes the place where the transcription is supposed to start from. Does it leave or it still continues with the rest of the polymerase, with the rest of the coenzyme?
That's a very good question. So its job is to identify the promoter. So once it identifies the promoter, it doesn't have any more use. So if the polymerase wants to move, the sigma factor falls off. We call it promoter clearance.
Oh, okay. All right. And then my follow-up question is that you also said that it has a helicase activity too. So I'm a little bit confused. Since it falls off, how is it able to undertake the helicase activity also?
That's also another very good question. Now, I hope you know that there can be more than one helicases, right? For
Yeah.
instance, if you take the helicase activity in eukaryotes replication, we know it's MCM. That
Yeah.
is the mini chromosome maintenance complex. That complex comprises of several subunits. So if one complex is not there, another component of the complex can carry it out. But in this RNA polymerase case, the helicase activity, you remember, let's go back to that slide. I didn't really focus on that because it's a minor activity. If it's not there, elongation can still go on, but for the local melting, we'll see that here shortly. Local melting, that is the, over here, yes, the local melting. Can you see?
Yeah, I can see that.
Initiation. So for local melting to be carried out, you need the sigma factor. You need the helicase activity of the sigma factor. But going forward from there, when it drops off, the polymerase is able to use other complexes to provide helicase activity.
Oh, okay. All right.
All
I
right.
get it. Thank
Thank you
you.
very much. All right, sure. So we were somewhere here. Yes. Is there any further question? Any more questions? Um, the questions help. Yes, there's someone asking a question. Victor, Victor AL, do you have a question? Madam Coswell, I think I should mention things and ask questions.
You should mention what.
I'm seeing very nice names here. I think I should mention some and then ask
Yes, please.
questions. So I mentioned Victor AL. Victor, if you're there, just give me a hi. If you give me a hi, we'll proceed. If not, I'll ask a question to you.
He has raised his
I mean,
hand.
a verbal hi, not the hand hi. Okay, maybe Victor is busy. So let's go on. We have just about 30 minutes and transcription is quite a lot. So if there's no question, this is the very basis that going forward you have to be able to recall from here. Because when we enter activators, inducers, repressors, and enhancers, it's going to be a whole different ball game. So if you are sure you understand this portion, then let's go on. Okay. All right. What this first point is trying to say is that transcription and translation don't occur at the same time in eukaryotes. Transcription occurs in the nucleus, whereas translation occurs in the cytoplasm. And since the eukaryotic cell structure involves compartmentalization, you can't have translation going on simultaneously as transcription. Transcription will have to end. The mature mRNA will have to travel from the nucleus through the nuclear pore into the cytoplasm for the tRNAs to start translating. So that's a very important point you have to know. It's a good to know point because you wouldn't be asked this question directly, but you'll be asked questions which require that you apply your knowledge of the fact that eukaryotic translation is not coupled to transcription. One happens before the other. both in the place they occur and at the time they occur. All right. In eukaryotes, there always has to be post-transcriptional modification, in that the nascent RNA is not, so we have modern methyl.
Aziz,
Hi.
please, your line was breaking when you
Oh,
were
okay.
talking about the post-transcriptional modification.
Okay, so we have three major types of post-transcriptional modification. We have the addition of the cap or the 7-methyl-guanosine cap. We have splicing, and then we have polyadenylation, which the layman calls tailing. So these are the three major processes that go on during post-transcriptional modification. And they are all very important because they make the mature mRNA, or they process the nascent RNA and make it mature and suitable for reading by the tRNA. They also protect the mRNA from exonuclease activity, and then they enhance its transportation from the nucleus to the cytoplasm. You know, the conditions in the nucleus are quite different from those found in the cytoplasm. So it has to be protected with a 5' cap, the helmet. It has to be protected with a polyadenosine tail, the shoes, and then it has to be spliced to be made tough. All right. So the most important, yeah, this is also a very high yield point. I have to admit, yeah, this is a very high yield point. You have to know what is transcribed by what. As for this, is very simple, very, very simple. So you have to know that eukaryotes have three types of polymerases: polymerase 1, polymerase 2, and polymerase 3. Oh, okay. Okay, Victor, I get you now. I think Victor had a problem with his microphone, so he couldn't talk. So eukaryotes have three types of polymerases. These are the three polymerases, and each of these polymerases transcribe a specific type of RNA. So polymerase 1 transcribes ribosomal RNA. Polymerase 2 transcribes messenger RNA. Polymerase 3 transcribes transfer RNA. Okay, and then note the exceptions in the brackets. So polymerase 1 transcribes all ribosomal RNAs except the 5S RNA. That 5S RNA is transcribed by tRNA, polymerase 3. So that is the very important thing you have to know. Let's go on. Yes, promoters. Remember that we said that genes don't have to be expressed at all times. They have to be expressed at specific instances. And so because of that, there's something called regulation of gene expression. So regulation of gene expression is carried out at different levels. The first level at which gene expression occurs is at the promoter level. So whether the promoter is good enough or bad enough allows for the specific expression of a particular gene. Remember over here, we said that the gap between the 35, the minus 35, and the minus 10 sequence has to be exactly between what? 16 and 19 base pairs. So if this is not right, it can affect the rate at which transcription will occur. If this sequence is not TAT, AAT, and it's like TGT, CCT, when the polymerase gets here, it wouldn't feel motivated to go and transcribe. So by by that way, you have affected what? Gene expression. The same thing applies for the -35 sequence. And remember, in prokaryotes, this sequence is called the Pribnow sequence, Pribnow, Pribnow sequence. P for prokaryotes, P for Pribnow. In humans, it's called the Hogness sequence, Hogness. H for humans, H for Hogness, P for prokaryotes, P for Pribnow. All right. So that is the first layer of gene regulation, the promoters. Promoters is a whole topic, so please pay attention at this point. Initiation of RNA synthesis at random points in the molecule will be a waste of energy. In fact, it will be a very significant waste. But the promoters serve as a guiding sequence that direct the polymerase enzyme to the initiation site. So promoters are the first level at which gene expression is regulated. First level of gene expression is the promoters. And you have to know this also. This is a high yield point. The promoters are read by the sigma factor in prokaryotes. However, in eukaryotes, they are read by the GTFs, the general transcription factors. So you have to know this, the difference between the sigma factor and the general transcription factor. They all read the promoters, but they are found in different organisms. So this is one major difference between eukaryotic transcription versus prokaryotic transcription. The first one we mentioned was that prokaryotic transcription is polycistronic. Eukaryotic transcription is monocistronic. So we have two differences now. As we go on, we have to keep note and then get more. There should be at least 10 differences between prokaryotic and eukaryotic transcription. The next thing over here, we have seen this diagram already, but you see that there's a color change in this particular one, which shows us that the RNA polymerase has now, by the help of the sigma factor, has now seen the -35 sequence. So now the RNA polymerase is smiling. It knows that there's an initiation point nearby. And then immediately it gets here, it begins to smile wider because then it knows that, oh, we are very close. And once it gets here, it forms the closed complex. So we are still on the promoters. This is just English. But the most important thing you have to know from here is that this point. So let me give you a second to read it. Just this one. The efficiency with which an RNA polymerase binds to a promoter and initiates transcription is determined by these sequences and spacing between them and their distance from the transcription site. It's very important to the control of transcription. All right. So we are on the stages of transcription. There are 3 stages of transcription. There are 3 stages of translation. So IET, initiation, elongation, termination. You will meet the same thing when we get to translation. Initiation comprises of several steps. The first step is the formation of the closed complex. But when you form the closed complex, it's like you are trying to enter a room and the door is closed. Sorry about that. You are trying to enter a closed room, that what I'm trying to say is that it's highly unstable. It's not possible because the complex is closed. The double strand is closed. So the closed complex comprises of the holoenzyme on the promoter. So this is a closed complex. RNA polymerase holoenzyme minus 35 sequence is a promoter sequence. There's nothing else going on. You just have the promoter and you have the holoenzyme. That is a closed complex. The next thing that has to happen is that the complex has to be opened. So that is the next point, open complex formation. So once you open it, the complex becomes more stable. And once it's more stable, then transcription can commence from there. How does it become stable? It becomes stable by melting the double strand. It becomes stable by melting the double strand. Once it melts the double strand, the complex becomes more stable. Now the next thing is like what most of the guys have been doing to the ladies nowadays. They tell you that before you get married, you have to try to what? Do certain things to find out if the person is the right person for you, and whatnot and whatnot. Well, these things didn't start from human beings. RNA polymerases were doing that before we even thought about it. So RNA polymerase will do what we call the abortive cycles. It will synthesize and see if that's the correct reading frame. So it will synthesize about a few nucleotides and then stop. Synthesize a few and then stop. What it's trying to do is that it's comparing all of those 100 oligonucleotides, trying to see if they are the same. Once it's convinced that, okay, then indeed this is the +1 site and this is the open reading frame, then elongation can start. I'm not encouraging the guys to keep on doing that. At a point in time, you have to be like the DNA polymerase. The DNA polymerase does not have abortive cycles. All right. So elongation. Elongation starts immediately the polymerase enzyme is cleared from the promoter sequence. That's what we call it, promoter clearance. So once the DNA, the RNA polymerase is able to move or begins to move, let me use the correct word. Once the RNA polymerase attempts to move from the promoter, then we are entering what we call promoter clearance. And the RNA enzyme can only move from the promoter when the sigma factor falls off. Until the sigma factor falls off, the RNA polymerase enzyme can never move from the promoter. So the gentleman who asked the question about the sigma, this is the answer to the question. The sigma only identifies and is done. After it identifies, it goes away, and then the core enzyme continues elongation. Why is it called core enzyme? Because it has lost the sigma factor. And you know the sigma factor makes it a holoenzyme. So once you lose the sigma factor, it's no more a holoenzyme. It's now a core
50%.
All right. You have to know that.
Aziz,
Hi.
please can you mute the people, the other people in the background? This thing is disrupting the lecture.
Okay.
Thank you.
You're welcome. So it's like Joseph of Arimathea. The only thing he did in Jesus' life is to provide him with a tomb. That is the function of the sigma factor. It's just to identify the promoter sequence and then bind to the promoter sequence and then form the closed and open complex and then abortive cycles. Immediately that is done, its job is finished, and elongation proceeds. Now, elongation is characterized by increased processivity. What does this mean? When elongation is set in motion, the RNA polymerase begins to speed faster than during the abortive cycles. That is an advantage of the promoter clearance. The next thing is that the polymerase enzyme moves progressively from the 3' end of the template towards the 5' end of the same template. And then there's rewinding of the double helix behind it. Then, of course, there's termination. Now, termination is high yield because there is an enzyme there, and anytime there's an enzyme, there's a possibility for an enzyme defect. And once there's an enzyme defect, then you need a doctor. Sorry, before you need a doctor, there will be a disease. And once there's a disease, you need a doctor. And once you need a doctor, you look for a medical student. So termination occurs in two types. Oh, Mary Jo, sorry. Welcome. We have two types of termination. We have the row-dependent termination, which uses an intrinsic sequence of GC inverted repeats, followed by intervening nucleotides and then AT-rich repeats. So when the polymerase transcribes the GC inverted repeats, the intervening sections, and the AT repeats, it forms a loop. And that loop cannot be held by the RNA polymerase. The RNA polymerase does not have the structure for holding a loop. So this is how the loop forms. It's like a hairpin and then down. So let's say the mRNA was synthesized in this way, and then when it gets to that place, you know that the bond between guanine and cytosine is very strong because it's 3 bonds. Guanine and cytosine form 3 bonds, but adenine and thymine form just 2 bonds. So you see that anywhere there has to be an opening, for instance, in the Tata box, there has to be an opening. So you find that the gene uses thymine and adenine because it knows that an enzyme will surely come and open there. So let's not make things hard for the enzyme. So they use thymine and adenine for that. But where it is expected to be very tight, they use guanine and cytosine. So this region here that I have drawn like a ladder is very strongly bound. There's nothing that can open it, which suggests that it is made up of what? Guanine and cytosine bases. Over here, you could have other bases. So the key thing you have to know is that you have guanine cytosine rich inverted repeats followed by intervening nucleotides, then AT rich sequences, which forms a hairpin structure which cannot be bound by the RNA polymerase pocket because it does not have a pocket for it, and so it dissociates and releases the nascent RNA. That is row independent termination. But when we come to row dependent termination, it's very simple. It uses an enzyme called a Rho protein. So the Rho protein, RHO over here, has helicase activity. So the helicase activity just pulls the nascent RNA from the polymerase, and that's all. It's a no-nonsense guy. Sometimes the Rho protein is added to the row independent termination to make it more efficient. So sometimes don't think that if you have row dependent, you don't have row independent. Or if you have row independent, you don't have row dependent. Some genes utilize both row dependent and row independent, depending on how efficiently they want to terminate transcription. Any question on the stages of transcription? Emanuela Boateng, please, let's hear your question.
Please, can you explain the termination again, particularly the role independent?
Okay, gladly. So, the rule one. Okay, so let's see if I can use this diagram. So now we have I'm trying to explain the termination again. We have transcription going on, but let's say when we get here, from this portion, we start to see a series of genes, Cs, genes, Cs, and then other nucleotides, then A, T, A, T. This sequence, when transcribed, forms this structure, a hairpin structure. When it is transcribed, sorry, it forms a structure like this. So it's coming, when you get to the GC sequence, it forms a hairpin structure. This is the intervening sequence. Then this is the GC. Then you have AT. So the polymerase enzyme, when you look at the shape of its mouth, it's not fitted to hold this weird structure here. So once this weird structure comes in, it says, well, I've had enough. Then it opens up and everything falls off. That is the row independent termination. But with the row dependent, there's an enzyme here called row. When it sees that the polymerase has reached the termination, it just comes, then yanks the mRNA off the template, and its job is done. So the row termination is very straightforward. I believe you answered.
Yes,
Come on.
please.
Okay. There was a hand up. There was a hand up. The name was I think Boating. Okay, I'm sure when you're ready. Yeah. Okay. Anytime you're ready, just interrupt and then ask your question. So yes, this is a very important point you have to know, transcription factors. Transcription factors are the second level of gene expression. The first level was promoter level. The second level is transcription factors. We have two types of transcription factors, the general transcription factors and the specific transcription factors. You find the general transcription factors in eukaryotes, and then you find the specific transcription factors mostly in eukaryotes, but you also find them in eukaryotes as well. The interesting thing about general transcription factors is that they are only found upstream of the promoter, sorry, of the initiating site. So if you should find any transcription factor downstream of the open reading frame, it is likely it is a specific transcription factor. But when you come to the specific transcription factors, they could be anywhere at all, and they include enhancers, inducers, activators, repressors, and all that. So that is one thing you have to know. So usually when you hear transcription factor 2A, transcription factor 2B, transcription factor 2H. They are the general transcription factors. But when you hear enhancer, enhancer, repressor, you know, those terms, they are usually the specific transcription factors. And you usually find the general transcription factors in the eukaryotes. Okay, there are several inhibitors of transcription. In fact, there are two. Rifampicin is a very important antibiotic because it's able to inhibit the protein synthesis or transcription in bacteria. And you know, bacteria causes tuberculosis, bacteria causes bacterial meningitis. So if you're able to stop this bacteria from transcribing their genes, at least you'll be able to cure yourself. So rifampicin together with isoniazid and other drugs are used in a TB treatment. But how does rifampicin help in inhibition of transcription? It does that in two ways. The first way is that it binds the beta subunit. And you know the beta subunit is responsible for phosphodiester bond formation. So if the phosphodiester bonds can't be formed, then you can't form your abortive cycles. And if you can't form your abortive cycles, the polymerase will not be sure that that is the correct sequence. And if the polymerase is not sure that is the correct sequence, there won't be promoter clearance. And if there's no promoter clearance, there can't be elongation. And if there's no elongation, transcription cannot take place. So by blocking the beta subunit, rifampicin is able to what? Mitigate the replication of TB causing bacteria or meningitis causing bacteria. It also does that the same inhibition by binding to the first two nucleotides. So it prevents the addition of the third nucleotide using steric hindrance. So it makes it very difficult for the third nucleotide to be bound. And if this doesn't happen, of course, initiation would not even be complete for you to even talk about elongation. So you have to know these two mechanisms by which rifampicin is able to inhibit transcription. It's a very high yield point, but not as high. But the second one is not as high yield as the first one. So actinomycin D, it just intercalates with the complex, the closed complex. So sorry, it intercalates with the DNA. So when the polymerase is moving, there's a blockage. The polymerase cannot move beyond actinomycin D. So it's like you are driving on a stretch of road and then you find that somebody has built his house on the road. Instantly, your traveling becomes a waste. Next point, regulation of gene expression in prokaryotes. Yes. So at this point, a lot of people may start getting confused. So what I'll do is that I'll go one step back. Transcription factors. I hope we all get up to this point. And then we have mentioned that they are the second point at which gene expression is regulated. That is using the specific transcription factors or the general transcription factors. And I mentioned that the general transcription factors are usually found in eukaryotes, whereas the specific transcription factors, for the purposes of this discussion, prokaryotes are the go-to organisms for specific transcription factors. So with this in your mind, let's quickly take an example of a prokaryote that is using specific transcription factors. We want to see how a prokaryote is being able to, play around specific transcription factors to regulate the transcription of specific genes within itself. And that prokaryote we are going to look at is a bacteria. And that bacteria is called Escherichia coli. Escherichia coli because a lot of research has been done into it, and scientists know a lot about E. coli, and so they can say more about E. coli than they can say about themselves. So this is E. coli. Now, E. coli uses glucose as its source of sugar. But E. coli is a very smart organism. It can adapt such that when there's no glucose, it can easily start using lactose. And I hope by now you know that lactose is a disaccharide of glucose and galactose. So the E. coli is very smart that, oh, okay, if there's no glucose, then I'll take lactose and break it down, throw the galactose away, and then utilize the glucose like nothing happened. That is how E. coli operates. And for it to be able to undergo this very complex reaction, it needs 3 enzymes. Those are the enzymes listed here. And you have to know these 3 enzymes, GPT or GTP, Ghana Textile Printing something, GTP, the mass mentioned people. The first one is galactosidase, the second one is transacetylase, and the third one is permease. From their names, you can tell their function. Galactosidase breaks down lactose into glucose and galactose. Sorry, galactosidase digests galactose. Permease makes the cell membrane of the bacteria permeable to galactose. Transacetylase, no one really knows what it does. It's also part of the enzymes. Now, before this transcription process can take place, okay, so what I'm going to do is I'm going to draw the lac operon for everybody. So just give me a second. Let me insert a blank page so I can draw the lac operon. Okay. So one lac operon coming right up. We have the non-coding strand, the coding strand, the template strand. Then we have the 3 genes over there. 1, 2, and 3. Let's call this galactosidase. Let's call this transacetylase. Let's call this permease. So these are the 3 genes that have to be transcribed. It means that this is the +1 region. So when the polymerase enzyme gets here to start transcribing these genes. Okay, but when there's glucose in the cell will not even allow RNA polymerase to get here. How does it do that? It has something we call the operator site. So the operator site has a repressor. So let's call this the operator site. Let's call this the repressor. Please, have I lost anyone yet?
Yes, please.
Where did I lose you?
From everything. You start with everything. Not everything, everything.
I
I
told
mean,
you.
when you start talking about the operator site.
Okay, those words I should have defined them first. Not your fault, don't worry, I have to define them. So what is an operator? An operator is a place is a sequence where a repressor binds. So let's say a wicked guy wants to get you. The place where he knows he'll get you is when you are walking alone in the dark. So the operator is the darkness. The repressor is the wicked person. So if the repressor wants to inhibit the transcription of that gene, it doesn't have to go and bind anywhere. It just has to go to the operator. Okay, let's just say yes, a judge and a plaintiff. If you have a case against me, you don't go to my ancestors in the grave to tell them. No, you go to the law court, and then the law court is going to invite me to come over there. So I remember quite recently, let's say I bought a plot of land, which there's a case, there's litigation. So the second person who has also bought the same piece of land will not waste his time coming to me to tell me, hey, this land is mine. No. He goes straight to the law court, go and lodge his complaint. So the law court is the operator. They are going to operate. Now my opponent is the repressor. He wants to repress me from developing the land. So the repressor goes to the operator to go and lodge a complaint. Then the operator will stop me from developing the land. Okay. So an operator is where the repressor binds. That is the most important thing. An inducer is what an inducer stimulates the synthesis of the gene. Or let me say, an inducer removes the inhibition that prevents the transcription of the gene. So let's say now that my opponent, the repressor, has gone to the judge, the operator, to stop me from developing, I will also go for my lawyer. My lawyer, who is the inducer, will go to the law court, the operator, and go and remove the repressor. I'm taking it again. The law court is the operator. The repressor is my enemy who wants to stop me from transcribing. But he can't come to me because I'm operating on a different level. So he will have to go to the law court, which is the operator. Then the law court, once they receive the complaint from the repressor, they tell me, hey, a gentleman has lodged a complaint against you on this plot of land. So stop doing anything on it till we have resorted the case. Then I also said, no, I need this land. I need to build. So I also go for a lawyer who is the inducer. So the inducer also goes to the same operator and suppresses the repressor and says, hey, you repressor, get out of here. So the suppressor leaves, the repressor, is removed from the operator. And then the operator now tells me, okay, your lawyer came to us and your lawyer has defended your case, so now you can carry on. So that is what I'm going to draw next. All what I have said is what I'm going to draw next. So I have my double-stranded DNA. I have the sequence for one gene, the sequence for another gene, and the sequence for another gene. And these genes are beta-galactosidase, trans-acetylase, and permease. Now, from this diagram, let's go back to this diagram. So you realize that this here is the coding region, the region that will be transcribed. And remember that I said that prokaryotic DNA is polycistronic. So you can have one coding region like this, coding for 3 different proteins. And that is exactly what we have here. This is the coding region that is coding for 3 different proteins. Are you clear at this point?
Yes, please.
Are you sure?
Yes, please.
Okay. So now, these 3 genes are waiting to be transcribed. But remember that these 3 genes are used to metabolize lactose. But if the cell does not need lactose, it doesn't need to transcribe these genes. So now let's assume that now the source of glucose is finished. So now E. coli has to wisen up and start transcribing the lactose genes. So what happens is that there is an operator. The operator I told you was the cut. But always there's a repressor there because the cell does not need to transcribe lactose because it is using glucose. So at every point in time, the repressor is present on the operator. Okay? Then, when it's time for lactose to be used because glucose is finished in the cell, the inducer, which is my lawyer, will come and bind to the repressor. When it binds to it, it will change its shape.